So when you break an arm, an X-ray gives you this really clean binary answer. You see that jagged white line on the film, and the doctor points to it and says, there it is, it's broken.
Right, it's undeniable.
Yeah, exactly.
Yeah.
But the deeper you step into the world of molecular biology and cellular immunology, that comforting precision kind of just vanishes.
Oh, completely.
Suddenly, the X-ray machine is useless, and we're looking at a diagnostic landscape that is incredibly murky. I mean, we can see the microscopic parts of a cell, sure, but not always how they actually fit together.
Which is incredibly frustrating as a researcher, honestly.
Right. And that is why today we are doing a deep dive into a really groundbreaking 2026 paper from the journal Cell. It's by Wang and colleagues, and it completely changes this dynamic. They've essentially built like a Google Earth for human tissue.
Yeah, and it completely reframes how we look at disease and immunity, because up until now, mapping a tissue was it was like looking at a satellite image of a city. You could see the buildings, you could see the cars on the street, but you had absolutely no idea what the people on the ground were actually doing.
You just see the dots.
Exactly, just dots. But this new technology doesn't just show us where the cells live. It zooms all the way down to the molecular level and shows us exactly who those cells are, like shaking hands with on a park bench.
Which is just mind-blowing. And to really anchor why this matters for you listening, I want you to imagine the last time you had a swollen tonsil from a really bad sore throat.
Oh, we've all been there.
Right. Structurally, that tonsil is a literal war room. It's where your immune system is actively training its troops to fight off whatever pathogen you just swallowed. But before this paper, scientists could only really see the isolated soldiers, the cells and the weapons they carried, which are the proteins and RNA.
Yeah, they couldn't actually watch them communicating and organizing in their natural battle formations.
Right.
To appreciate the leap we're talking about here, we really have to look at the 3 layers of biology at play. So first, you have the blueprints, which we call transcriptomics.
Okay, the transcriptomic.
Right. This is the messenger RNA or mRNA. It's basically telling the cell what materials to manufacture.
So it's literally the architectural drawing.
Exactly that. Then you have the building materials themselves, which is proteomics. These are the actual proteins constructed from those blueprints. They're the physical machinery of the cell.
Got it.
But, having a blueprint and a pile of bricks doesn't explain how a skyscraper stays standing in a hurricane. And that brings us to the 3rd layer, which is sort of the holy grail of spatial biology. The interactome.
The interactome. I just love how that sounds. It's basically the difference between having a printed guest list of everyone who attended a crowded party versus seeing a photograph of exactly who is hugging whom on the dance floor.
That's a perfect way to picture it. It's the actual physical connections, the protein complexes. And in biology, that physical connection is the spark for everything. A protein on the surface of an immune cell might just sit there totally inactive for days. But the millisecond, it physically touches a very specific protein on another cell.
It sets off a chain reaction.
Exactly. It triggers a massive cascade. It can tell a cell to multiply, to attack a virus, or even just to shut down and die.
Okay, let's unpack this. Because conceptually, the idea of mapping out every single handshake in a tissue makes total sense. We want to see the hugs at the party, but mechanically, I'm trying to picture how it is physically possible to see 2 microscopic proteins actually touching inside an intact human tissue slice.
It requires just sheer mechanical genius. The experimental tool is called Sprox-seq. That stands for spatial proximity sequencing.
Spatial proximity sequencing.
Right. And the chemistry of how it works is brilliant. They start by taking a slice of an intact human tonsil that is only 10 micrometers thick.
So incredibly thin, basically a single layer of cells.
Yes, exactly. And they place this slice on a special capture surface called a Visium slide, which is going to be crucial later. Then they flood this tissue slice with AOCs.
AOCs?
Yeah, antibody oligonucleotide conjugates.
Which is a massive mouthful. I mean, when I was reading the paper, I found it easiest to think of these AOCs like homing pigeons.
That is a fantastic analogy, actually. The pigeon itself is an antibody, and we can train that antibody to fly to and land on one very specific protein.
Right.
But tied to the pigeon's leg is a tiny, unique barcode made of DNA, and that's the oligonucleotide.
Okay, so if I want to map a protein called CD19, I release millions of these CD19 pigeons, and they're all carrying a specific barcode tied to their leg.
Yes, and you release them in specific pairs, probe A and probe B. Now, if 2 target proteins on a cell surface are sitting incredibly close to each other, and we are talking about a microscopic gap of like 50 to 70 nanometers.
Wow, so basically touching.
Exactly. If they are that close, the DNA barcodes from probe A and probe B will physically reach out and link up. They literally ligate together. They perform this permanent chemical handshake.
Wait, let me challenge that for a second.
Sure.
If I can send a bunch of probes into a tissue, and I can just look at my slide and see a massive amount of CD19 and another protein in the exact same microscopic spot,
Yeah.
can't I just assume they're interacting? Like, why do we need this wildly complex handshake chemistry at all?
Because of the resolution limit of our tools. Even our absolute best spatial transcriptomic microscopes are, well, it's like looking at a crowded stadium from a blimp.
Okay.
You might see two people standing in the same square meter of the bleachers. But are they shaking hands? Or are they just facing opposite directions, completely ignoring each other?
Oh, I see.
Yeah, in a crowded cell membrane, proteins are just packed together. Just being in the same neighborhood doesn't mean they're functionally interacting. The probes absolutely have to physically link up or ligate to prove an interaction is genuinely taking place.
That makes so much sense. But then, how does the slide actually read this handshake? I know the Visium slide they use captures RNA because natural mRNA has this thing called a poly-A tail, right?
Right, yeah.
It's a long string of A nucleotides at the end. And the slide essentially acts like a grid of microscopic magnets that pull down anything with a poly-A tail. But our homing pigeon barcodes, they don't have that tail.
Right, and this is where the engineers got incredibly creative. Probe B is engineered with 6 specific nucleotides at the very end called UTPs.
UTPs.
Yeah. Think of these UTPs like chemical masking tape. After the probes link up and form that handshake, the scientists wash the tissue with a highly specialized USDR enzyme.
Like microscopic scissors.
Yes, scissors that only look for that specific tape. The USDR enzyme recognizes those 6 UTPs and snips them right off. And hiding directly beneath that tape is an artificially engineered poly-A tail.
That is just brilliant.
Isn't it?
So suddenly, this brand new piece of DNA, this chemical proof that two proteins just shook hands, now has a tail that makes it look exactly like normal cellular mRNA.
Which means when they activate the capture process on the Visium slide, the slide pulls down the cell's natural mRNA and these artificial handshake products simultaneously,
Right.
and it preserves their exact geographic location in the tissue.
Even with that, though, inside a crowded cell membrane, proteins are just chaotic, right? They're bumping into each other all the time. How do the researchers know it's a real purposeful handshake and not just an accidental bump on a crowded subway?
Right, the subway bump. They filter the data through heavy statistical algorithms, specifically one called Fisher's exact test.
Okay.
It's a mathematical filter that calculates the probability of 2 proteins being next to each other by pure random chance. If the probability is too high, the algorithm flags it as background noise, an accidental bump, and throws it out.
Got it.
And if it passes, they calculate a protein fractional overlap score, which grades the actual intensity of the interaction by adjusting for how abundant the protein is. Because if a protein is literally everywhere, you'd expect more accidental bumps, so the algorithm accounts for that. It leaves you with a map of pure, verified biological affinity.
Okay, so let's do a quick mini recap for you listening and make sure all these concepts are locked in. Sprox-seq uses barcode-carrying antibodies, those homing pigeons.
Yep.
It uses proximity ligation to capture interactions with that masking tape and scissor trick. And it uses statistical algorithms to verify them, all while preserving the tissue.
Beautifully summarized.
So my question for you, the listener, to sort of mull over is, if you could map the molecular handshakes in any organ in your own body right now, which one would you look at first?
Oh, that's a great question.
Right. Now, moving on, now that we have this incredible mapping tool, the researchers pointed it at the ultimate immunological test case, which is the tonsil war room.
Yes, and to really read the map they created, we need to understand the geography of the tonsil. Sprox-seq successfully mapped all the distinct anatomical zones, but the most critical area is the germinal center.
A boot camp.
Exactly, the actual boot camp where your B cells learn to fight off specific pathogens. And this boot camp is divided into 2 distinct environments.
Right, so first is the dark zone. And biologically, the dark zone is like a massive library.
Yes, very quiet, very intense.
The B cells here are furiously mutating their genetic code.
Right.
They're trying to randomly generate a better antibody to fight the virus you just caught.
Yeah.
They're studying, they're multiplying, but they're relatively isolated.
And then they leave the library and migrate to the light zone. This is the testing arena. The B cells are confronted by specialized instructors called follicular dendritic cells, or FDCs.
Right.
The FTCs essentially interrogate the B cell. They test the new antibody to see if it actually works.
Here's where it gets really interesting. When the researchers ran the Sprox-seq protocol, they mapped 32 different surface proteins and a staggering 528 pairwise interactions across the tonsil.
It's a massive amount of data.
Huge. And when they generated the interactome map, the light zone lit up as this massively dense, incredibly complex protein interaction network, while the dark zone was relatively dim.
What's fascinating here is how perfectly that network complexity mirrors the underlying biology. The light zone is a hub of extreme life-or-death communication.
Right.
B cells are constantly bumping into FTCs, fighting with other B cells for space, and desperately seeking survival signals. If a B cell's newly mutated antibody fails the test, or if it doesn't get the right physical handshake from an instructor, it triggers apoptosis.
It just dies.
Yes, the cell dismantles itself and dies.
And looking at this data, it really changes how I view my own body. We think of our tissues as these static things, but there are billions of microscopic interrogations happening in our throats every time we get the sniffles.
Billions, constantly.
It's like the difference between sitting quietly in a cubicle versus fighting to be heard in a high-stakes board meeting. The light zone is the boardroom.
And Sprox-seq didn't just capture generic noise in that boardroom either. It identified highly specific canonical protein complexes that immunology tells us should be there, which really proves the platform works flawlessly in intact human tissue.
Okay, time for our second mini recap. So the tonsil features distinct zones. The dark zone is the quiet library, but Sprox-seq proved that the light zone is this highly complex, interactive boardroom network.
Exactly.
So for you listening, think about your own workplace or community. Where is the biological light zone in your life where all the critical networking and communication actually happens?
I love that framing.
Yeah, it helps visualize it.
Yeah.
But, the tonsil isn't frozen in time. The cells are constantly moving from the cubicle to the boardroom. How do we see that progression? Like, how does a cell transition from a naive recruit to a seasoned, battle-tested veteran?
So in spatial biology, researchers rely on a mathematical concept called a pseudotime trajectory.
Pseudo-time trajectory.
Right, because we can't put a tiny video camera in your tonsil and track a single cell aging over time. We use the massive amount of data captured in the slice to infer a timeline. It's like taking 1 million photographs of different hikers all along a mountain trail, and then using the background scenery in those photos to piece together the entire path, from the base camp all the way to the summit.
But the researchers found a massive twist when they tried to build this trail map, though. When they looked purely at the RNA blueprints, the transcriptomics, the algorithm suggested one relatively simple timeline for how these immune cells matured.
Yeah.
But when they built a trajectory using the protein interaction data, the actual handshakes, it revealed a completely distinct biological path.
And the interactome mapped a far more accurate functional path. It started from the outer edges, the non-GC follicles where naive cells live. Then it moved deep into the dark zone library and finally culminated in the light zone testing arena.
Right.
They tracked specific protein complexes along this exact timeline. For instance, they looked at a handshake between two proteins, CD19 and CD21.
And they found this specific CD19-CD21 complex was highly enriched in the naive B cells before they ever even entered the boot camp.
Exactly. But as the cells moved into the germinal centers, a completely different handshake took over, which was the CD21-CD35 complex.
And later, as the cells survived the entire testing process, a complex called CD20-CD32 became highly visible, linking directly to mature memory B cells.
Right.
But wait, if I just measure the amount of CD19 floating around in a cell and the amount of CD21 floating around, doesn't that tell me they're interacting? Like, why was the RNA trajectory so fundamentally different from the interactome trajectory?
Well, having the ingredients on the counter does not mean the cake is baked.
Ah, I like that.
You can have an abundance of CD19 and CD21 proteins just sitting on the surface of a cell doing absolutely nothing.
Yeah.
The transcriptomics only tell you the cell possesses the building materials.
Right.
You have to measure the interaction directly to understand what the cell is actually experiencing at that exact moment.
Which brings up the ultimate question. What is that handshake actually doing?
This raises such an important question, and it's really where the brilliance of Sprox-seq capturing both RNA and protein interactions on the exact same slide pays off.
Because they overlay them.
Exactly. Because they had the blueprints from the exact same microscopic spots where the handshakes were happening, they could look at what the genes were doing, precisely where the CD21-CD35 complex formed.
And they found a direct correlation to mitotic pathways, cell division.
Yes. In the exact geographical spots where CD21 and CD35 were physically linked, specific genes like PLK1, CENPF, and E2F targets were suddenly running at maximum capacity.
Now, for those of us who don't dream in acronyms, genes like PLK1 and CENPF are essentially the mechanical engines that physically pull a cell's chromosomes apart so it can split into two.
Exactly. The researchers directly linked a physical protein handshake on the outer surface membrane of the cell to the internal genetic command center, telling the cell to fire up its engines and multiply.
It's incredible. It's like a molecular secret handshake to enter a very exclusive club. The cell doesn't even know it's supposed to divide until it gets that exact physical touch.
It's beautifully coordinated.
It really is. Okay, time for recap number 3. Protein interactions reveal cell maturity trajectories that RNA totally misses. And specific complexes like CD21, CD35 directly correlate with cells actively dividing.
Nailed it.
So to you listening, does it change how you view your own biology, knowing that parts of a cell are turning on specific genes only when they are physically holding hands with another specific protein?
It's a trippy thought, honestly.
It really is. But up until now, we've only been talking about proteins holding hands on the same cell.
And that is a crucial distinction. In biology, a cis interaction, CIS, is when two proteins on the membrane of the same cell connect with each other. It's basically like clasping your own hands together.
But a trans interaction, TRANS, is when a protein on one cell reaches across the biological void and physically locks onto a protein on a completely different cell.
Like reaching out and shaking hands with your neighbor.
Right. And to truly understand the sheer complexity of the light zone, the researchers had to prove they can map those trans interactions too.
Yes, and they focused on a classic biological case study for this. The interaction between a protein complex called VLA4 and a protein called VCM1.
So VLA4 lives almost exclusively on the surface of the B cells, the recruits. And VCM1 lives almost exclusively on the surface of the follicular dendritic cells, the FDCs, which are those harsh instructors.
Right.
So since these proteins live on two entirely different types of cells, any connection between them must be a trans interaction. It has to be a bridge between two distinct entities.
And Sprox-seq directly captured this VLA4 to VCM1 physical contact happening right in the light zone.
It's like we didn't just map the cities and the roads on Google Earth. We literally photographed a molecular car crossing a physical bridge between two cellular states in real time.
And the biological implication of that bridge is profound, honestly, because when they looked at the RNA in the spots where this connection formed, they didn't see massive changes in gene expression like they did with the cell division example.
Wait, why wouldn't genes turn on? If two completely different cells are connecting, shouldn't alarms be ringing?
Because the purpose of this specific interaction is structural, not genetic.
Oh, structural.
Yeah. The light zone is a very chaotic, fluid environment. This trans interaction acts as a primary adhesion pathway. It is a literal physical anchor.
Okay, I see.
It keeps the B cell physically tethered to the FDC long enough for the instructor to test the antibody and deliver the necessary survival signals. If that anchor breaks prematurely, the B cell just drifts away into the void and dies.
Wow. So it's not a handshake, it's a molecular seatbelt.
A molecular seatbelt, that's perfect. And Sprox-seq proved we can map these seatbelts by the tens of thousands across an entire slice of human tissue.
All right, our final mini recap here. Sprox-seq can detect trans interactions between entirely different cells, essentially mapping physical anchors like the VLA4 to VCM1 seatbelt in intact tissues.
Spot on.
So, question for the audience. What other vital hidden bridges do you think could be discovered using this technology in diseases like Alzheimer's or autoimmune disorders?
The applications there are just virtually limitless. We are entering an era where we can map the physical architecture of disease at the most fundamental level.
So what does this all mean? When you pull back the lens, how do you synthesize what Wang and his team have accomplished here?
If we connect this to the bigger picture, Sprox-seq has added a brand new vital dimension to spatial biology. We aren't just looking at static lists of cellular parts anymore.
Right.
We aren't just reading the blueprints or counting the bricks. We are watching the biological machine run.
Yeah, in real time.
Exactly. By mapping these exact communications, the cis and trans interactions, we are decoding the physical language that drives immunity, tissue organization, and cellular survival. It proves you cannot fully understand a biological system without understanding its physical relationships.
It's basically the difference between memorizing the vocabulary of a foreign language and actually sitting in a cafe listening to a fluid conversation.
That is perfectly said.
I want to leave you, our listener, with a final provocative thought. something that builds on this extraordinary leap forward, but wasn't explicitly answered in the paper. We've just seen how Sprox-seq can map the exact surface interactions that keep immune cells alive or trigger them to divide.
Right.
What happens when we apply this Google Earth of proteins to cancer tumors?
Oh, that is the absolute frontier of oncology right now.
Because could we soon use this technology to scan a tumor and find the exact hidden molecular handshake that allows cancer cells to cloak themselves and hide from the immune system?
Yes.
And if we can find that specific toxic embrace, could we design a targeted drug to simply step in and block it?
I mean, it's not just a possibility, it's the inevitable next step. We will find those cloaking mechanisms and we will break them.
It's an incredibly hopeful place to leave off. Thank you for joining us on this custom-tailored deep dive. We hope this exploration reminds you that true understanding comes not from drowning in data overload, but from finally seeing how all the individual pieces connect. Because sometimes, to understand the disease, you just have to look at who is shaking hands.